Evening everyone, and welcome to our live stream for the second biology paper. So that's B4, B5 and B6 on the OCR gateway. For those of you doing AQA, then what you're going to find is that while we do have certain topics that are going to overlap, particularly for the first part of the live stream, when we get to the very end, then that's something you've already done your paper one. So it's all of OCR gateway for those of you doing combined and triple foundation and higher. If you're doing one of the other exam boards, it's a bit sort of piece it together and there will be bits that work for you. So what we're actually going to start off with then is B4, which is all about the ecosystems. So first thing we need to know then are a few of these key terms that are really important for you to understand when we're looking at the ecosystems themselves. So an ecosystem is all of the living organisms and the physical conditions in an area. The community are the organisms within the ecosystem itself. The habitat is the area in which the organisms live. And then we've got some words which we associate with the food chains as well. So we've got the producer, which is any organism that makes its own food via photosynthesis. So that would be things like the plants and the algae. We've got the consumers, which are the organisms that don't make their own food, and therefore they have to eat other organisms to get that energy. So any animal really you can pick for that one. And then we've got the decomposers, which are the ones that gain their energy by feeding on any of that dead or decaying matter. So that would be your bacteria and your fungi. For those of you doing the GCSE biology, then you do need to know about those food chains. So hopefully we remember that when it comes to food chains, then one of the most important things is the direction of the arrow itself. So the arrow is going to show you how the energy is moving through that food chain. So when we're drawing it, if you've got grass and a rabbit, then the energy goes from the grass to the rabbit. So the arrow has to go that way. It can't go that way. Otherwise, what you're saying is energy is going from the rabbit into the grass, i.e. the grass is sitting there devouring rabbits when they're least suspecting it. Doesn't happen. Now, what we actually find is the ultimate source of all energy is going to be the sun itself. So the plants take in the energy from the sun through the process of photosynthesis. So the energy is absorbed by the chlorophyll, and then that process of photosynthesis takes place to make the glucose. This could be a point where they touch back on the work in B1, where obviously you would need to know the word and balance symbol equation for photosynthesis. So carbon dioxide plus water makes glucose and oxygen. And remember, to balance it is dead easy. Just stick a 6 in front of everything that's not glucose, and then you've got it right. Once we've made the glucose, the plants will then change that into carbohydrates and proteins within the plant itself, and it becomes part of the plant's biomass. So that when animals then come along and eat the plants, biomass gets transferred from the plants to the animals, sorry, and that means that they're then going to build that into their own biomass. We don't, however, transfer everything from the plant to the animals. And the reason for that is that some of the energy will be lost through various processes like respiration. Now, I've mentioned that phrase biomass a couple of times there, and biomass, quite simply, is the amount of living material that's present. So when we're actually looking at this, what we're actually saying is we're looking at the transfer of biomass in one of these food chains. And we're looking at the biomass that's transferred between what are called trophic levels. And a trophic level, quite simply, is a feeding level. So it just shows the position of an organism within that food chain or food web. And remember, in any given habitat, then we've got lots of food chains that all link together in that big food web. Back to something that you need to know for both the GCSE biology and the combined science, we need to know about the difference between biotic and abiotic factors. So the biotic factors are the living factors. So this is things like how many organisms there are, what organisms they actually are, and any competition between them. So they're all living factors, so biotic. The abiotic factors are the non-living factors or the physical factors. So this is things like the temperature, the pH, the amount of rainfall. All of those are non-living, so they're abiotic factors. What we need to understand then is how these abiotic factors will influence the different organisms that live there. So if we think about one of the common ones, first of all, light. So light we know is required for photosynthesis, and the greater the light availability, then the greater the success of the plant is pretty standard. And what we actually find, though, is it's not a case of plants can only grow in areas where there are massive amounts of sunlight. Even in those shady zones, some plants can exist. The key thing that's happened there is that process of evolution, which we're going to touch on in B5. What we see is those plants have evolved to actually cope in that low light intensity. So they've done things like grow really large surface area leaves in order to absorb as much of that sunlight as possible. Second abiotic factor is temperature. Now, temperature is important because it's all about enzymes. Again, link back into your first paper here. Hopefully we do remember that enzymes are going to be quite seriously affected by temperature, so that initially as you warm them up, then there's more frequent successful collisions, therefore a greater rate of reaction. But as we go beyond that optimum temperature, then what happens is the enzymes get denatured, the active site changes shape, and it no longer works. So temperature is going to affect any enzyme-controlled reaction inside an organism there. And what we'll find is there are certain organisms, like lizards, for example, that aren't warm-blooded themselves. Okay, they're cold-blooded or ectotherms. And that means that in the morning, if you ever see them, they all creep out of wherever they've slept overnight, and then they'll find a lovely sunny rock, or if you're in a hotel, the main roads and paths, because they're nice and warm, to then warm themselves up and get their metabolic reactions happening faster. Third one, all about water. Hopefully we do know if you don't have enough water, then you die. It's pretty standard in any living thing. If they don't get enough water, it can lead to death. And remember, the first stage in plants is they're normally growing nice and upright. If they lack water, then they start to wilt because the cells are no longer turgid. Last factor we're going to look at is pH, which is again going to link into our plants primarily. Now, what we actually find is that the pH can actually affect what plants grow in your garden. So if you live in an area that's got quite acidic soil, then you're better off growing things like rhododendrons. Whereas if you've got alkaline soil, then you'd be better off growing things like a cucumber, because they're suited to the different pHs, which is why some plants just do terribly when you try to grow them in your garden there. The only other thing we need to concern ourselves with in terms of the abiotic factors is how we can actually measure them. So if you're carrying out any kind of investigation, then you need a way of measuring these abiotic factors, either to use them as a control and therefore say, we know this stayed the same and therefore it didn't affect our investigation, or to actually investigate how those changing abiotic factors do influence what lives there and how it grows. In terms of our light intensity, we use a light meter. For the moisture, it's a humidity sensor. pH is a pH probe. And for temperature, it's just a thermometer. So just bear those in mind. If they ask you how you could work out the temperature of the soil, you use a thermometer, okay? Our next little bit is coming on to competition and interdependence. Hopefully we do know that when we're looking at what plants and animals need to survive, then they need a range of different things from their surroundings. So plants will need water, light, minerals, space, all of those things. Animals are going to need their water, their food, they're going to need breeding partners, shelter, territory, anything like that. Now, within any given area, you're not just going to have one of each organism. So what we actually find is that if there's a particular material that they need that's in limited supply, then all of those organisms that require it are going to be fighting. They're going to be competing for those resources. So what we see as a result of that competition is the weaker one could just die, or they could leave the area and move somewhere where there's less competition. So it will influence what organisms can actually live there. And when we're talking about population, that's just the number of organisms of each species that exists in an area. And that competition will have a direct impact on the population size itself. When we talk about interdependence then, this is how different organisms all depend on each other within a given community. So what we find is we've got three main types of ecological relationship that we need to concern ourselves with. The first one is predation, which is that relationship between predators and prey. Now, what they actually have is quite a common question associated with a graph like this. So what we've got, the red line is our prey, which is a hare, and the blue line is the predator, which is a lynx. And they could ask you to describe this. Now, describe questions, it's say what you see. So on that one, you literally say that initially, then we see our predator line decrease, and then we see an increase in the prey not long after. That's the describe. That's all you need to do, is literally say what you see. If it was explain the graph, then you follow that same pattern of describing the shape first of all, but then you've got to back up why that happens using the science. So we see this decrease in the predators initially, followed by an increase in the prey, for the simple fact that if we've got fewer predators, then there's less of them eating the prey. So the prey numbers can then go up because there's fewer predators eating them. The reason we get that little bit of a delay between those two things is because it takes a while, obviously, for the impact to be seen. So if you're talking about things starving to death, that doesn't happen overnight. If you're talking about reproduction, therefore leading to an increase in population, again, gestation periods take a little while. So predation, first one. Second is mutualism, which is where both organisms benefit from this relationship. And the good example of this are buffaloes with the little birds, the oxpeckers that sit on them. So the buffaloes are providing that nice little safe patch that the birds will sit on them, and the oxpeckers, the little birds, are eating various parasites off the back of the buffalo. So they both benefit from this relationship. Third and final one is parasitism, which is where only one of the organisms will benefit, which is the parasite itself. The host, the one that's actually playing the host to this parasite, is going to suffer as a result of this relationship. And a good example here is the good old tapeworm. So the tapeworm will benefit by stealing every nutrient you put into your body. You as the host is going to suffer because you're not getting any of those nutrients. So 3 relationships: predation, mutualism, parasitism. For those of you doing GCSE biology, you need to know about the pyramids of biomass. Now, what we actually find here is the pyramid of biomass is the second type of pyramid, because the type of pyramid you probably saw lower down the school is pyramid of numbers. Now, as that suggests, it's a pyramid based on how many of each trophic level organisms are present. Now, what we'll find is you could have a couple of different designs of a pyramid of numbers, because what we find is we could have this one, where we've got lots of grass, for example, supporting fewer rabbits and then a very tiny number of foxes. Or we could have one like this, where we have a single oak tree supporting lots of aphids and a smaller number of birds. And that's the key thing with the pyramid of numbers, is it doesn't always form that pyramid shape. If we're talking about the pyramid of biomass, though, because that is the amount of living material that's present, it will always be pyramid shaped. So your pyramid of biomass will always be this pyramid shape. If they ask you to draw a pyramid of anything in your exam, then they are quite likely to give you a printed piece of graph paper. Now, that's there because you need to use a proper scale for the length of your bars, because the length of the bar represents either the number of organisms or the amount of biomass present. So you need to make sure that you've worked out an appropriate scale. You then plot the bars to the appropriate length to represent this, and then make sure you label the bars. So literally it could be as simple as just writing either on it or just off to the side what they represent. So all you'd have to do once you've plotted it is label the bars up with what they represent as well. As long as you've done that with the correct scale, and always start with the producer at the bottom, then you should get top marks on that should they ask you to do it. In terms of how we're going to calculate this biomass, we need to work out how much living material is present in each trophic level. So we take samples from each trophic level, and then we're going to calculate the average mass of the organisms from each. And the way that we're then going to do that is multiply up the average mass to the total population in the area to give us the total biomass of that given population. Now, in terms of working out the mass of these organisms, we use something called dry mass, because what we find is the water content within living things will vary. So what we actually do is we take it, we dry them out, usually in an oven, and then we record the mass of that. And that's the dry mass, which is what we then use to get our biomass calculation. As we said earlier, when we're looking at how energy gets transferred through a food chain, then not all of the energy that we start off with makes it to the end of that chain. Because what we find is at the very, very beginning, where we've got photosynthesis, then only about 1% of the energy from the sunlight actually makes it into the chemical stores within that plant. because an awful lot of the light that hits the leaf is reflected straight off, so it doesn't even get absorbed. Other parts there will be used for things like respiration as well, so we've got lots of energy losses occurring there. Once we've actually got that within our plant, then not all of the plant makes it into the animal. You don't often see creatures digging up every bit of the root to eat of the plant, so that's some of the biomass being left over. So every step of that particular food chain is going to lead to a reduction in the amount of energy being passed along. So what we actually find is that as a result of that, food chains are limited to 4 trophic levels usually, because of the energy losses we see. Beyond those 4 trophic levels, then we don't have sufficient energy to support life, hence why they only get to that 4th stage. The next thing is for everyone again, so combined and GCSE biology, which is looking at the different cycles. Now, hopefully we do know that for things to survive, they need various minerals. Now, what we find is this is in a natural cycle in our planet, because we've obviously got some of these minerals that are contained within living things, but when they die, then they get broken down and returned to the soil by the action of the decomposers. And a few key things that we find here, carbon particularly, which we're going to need in order to make fats, proteins, carbohydrates, DNA, pretty much any chemical you want to name in a living thing, and nitrogen, which we're going to use in order to make the DNA and the proteins in living things. The other one we're going to look at is water as well. So we're going to look at 3 cycles now: nitrogen, carbon, and water cycle. So first one we're going to look at is the nitrogen cycle. Now, the nitrogen cycle is pretty much about 4 types of bacteria. So we've got the decomposers, which are made up of bacteria and fungi. So if they ask you, name a decomposer, bacteria or fungi, that's what they're looking for. And they're going to be breaking down the proteins into ammonia. Second type of bacteria, the nitrifying bacteria, are going to turn the ammonia into nitrates. Third type, the nitrogen-fixing bacteria, turn the nitrogen into nitrates. And our fourth and final one, the denitrifying bacteria, turn nitrates back into nitrogen. So this is our nitrogen cycle. So we've got nitrogen in the air, which actually is pretty useless to plants. They can't take it in as nitrogen gas. So we have our nitrogen-fixing bacteria that convert the nitrogen into the nitrates, which can then be absorbed by the plant roots. That then gets changed into proteins in our plant. The animals will eat the plants and transfer into animal proteins. So we've now got proteins in both of our living things there. If they die, they undergo the process of decomposition as a result of our decomposers, which changes those proteins into ammonia. That ammonia can then be converted into nitrates through the action of the nitrifying bacteria, which again can be taken up by our plants. Remember, we also have the denitrifying bacteria, which are going to release nitrogen back from the nitrates themselves. So what we've got there is the basis of the nitrogen cycle, but it's all about the 4 bacteria: decomposers, nitrifying bacteria, nitrogen-fixing bacteria, and denitrifying bacteria. Second cycle, the water cycle. This one is just looking at how water moves around. So we start off in an ocean. Now, in our ocean down here, then what we're going to see is as the sun shines on it, we get evaporation. So the water that's as liquid form here turns into water vapor and rises up. As it rises, then it gets into the cooler levels of the atmosphere, and therefore it condenses into our clouds. As it continues to cool, it's going to then fall as precipitation, which is our rain. Now, that rain is going to either soak into the groundwater or it's going to run off the surface. And once we've got it in the ground, then our plants are actually going to take it up, and they will then release water back as water vapor into the atmosphere as a result of transpiration, which is the loss of water vapor from the aerial parts of a plant, as we learned previously. So the water goes around in that nice little cycle between the living things and the non-living parts of the environment as well. Third and final cycle is the carbon cycle. Now, the carbon cycle is looking at basically 4 processes. We've got respiration, decomposition, combustion, and photosynthesis. So we start off with carbon dioxide in the atmosphere first of all. Now, that carbon dioxide is only removed by one process, which is photosynthesis. So the only process that takes carbon dioxide from the atmosphere is photosynthesis in our plants. Once we've got that carbon in our plants, it's going to be used in order to build proteins, carbohydrates, etc. And we will then see a range of different things that could happen. First of all, it could actually die as a plant, and therefore we have decomposition occurring. We could see that it gets eaten by animals, and therefore it gets transferred into the animal itself. Or with any of these living things, whether it's bacteria carrying out the decomposition, the animals, or the plants, they're all carrying out respiration. And respiration releases carbon dioxide back into the atmosphere. The other option we've got is once things have died over millions of years, they could become buried, become the fossil fuels, which we then burn eventually. And through that combustion reaction, we release carbon dioxide back into the atmosphere as well. So what we've got, carbon dioxide being released into the atmosphere by respiration, decomposition, and combustion, and carbon dioxide removed from the atmosphere as the result of photosynthesis. So just remember the order of those, because sometimes they're giving you a basic diagram like that with the boxes that you've then got to put the processes in. When we think about carbon dioxide, it's not something that has a consistent amount throughout the entire day. When you actually see the graphs, they have this little spiky profile. And the reason for that is that the carbon dioxide levels will vary at different times of the actual day itself. Hopefully we remember photosynthesis can only happen during light, and therefore photosynthesis is happening only during the daylight hours for most plants. However, respiration is going to happen all the time. So that means that what we see, photosynthesis only happening during the day, but respiration happening day and night. So during the nighttime, we see an increase in the amount of carbon dioxide, which is not being actually removed because we're not having photosynthesis take place. Whereas it will then drop a little bit during the day because there is photosynthesis, which just keeps it a bit lower. But generally, if we have a look at a graph of the amount of carbon dioxide in our atmosphere on planet Earth, then it's increasing as the years go by. And there is quite a dramatic increase in more recent years. And that's all down to human activity. So the fact that our population has increased dramatically in recent years. And all of these people that we now have are obviously driving cars. We're all using electrical appliances which require energy, which comes from power plants. All of this contributes then to the increasing amounts of carbon dioxide in our atmosphere, something that those of you who do geography are probably really well versed in already. The next little bit we need to look at are the decomposers themselves. Now, decomposers are microorganisms, which, as we said, are bacteria and fungi. And their role is to break down or decay any of this dead organic material, as well as things like urine and feces. And what happens as a result of that process of decomposition is it's going to return the minerals back to the soil so they can be used again. One phrase that we use here is saprophyte. And a saprophyte is one that's going to feed on this dead material or waste by releasing the enzymes onto the surface of it. So rather than actually kind of having a mouth and eating it, what happens is they secrete the enzymes from themselves onto the surface of this material. The enzymes will then break it down, and then the organism absorbs the nutrients afterwards. So that's what a saprophyte is. Another type of organism we talk about here are detritivores. Now, these are small animals, and you're quite familiar with a lot of them, things like earthworms, woodlice, maggots. And what they do is speed up the process of decomposition by breaking this organic material up into really small pieces. So that what that then does is increase the surface area, and therefore we've got a larger surface area for the decomposers to work on, meaning it's going to get decayed much faster. If you're doing GCSE biology, then you do need to know the factors that can affect the rate of decomposition. And what we find here is if we want to increase the rate of decomposition, then we can increase the temperature to a certain extent, obviously not too high. We need it to be moist, so some actual moisture there, but not excessive amounts. And we need there to be plenty of oxygen available. The only other question I've seen them use here was looking at the rate of decay. And if they ask you to calculate the rate at which something is decaying, then just do the change in mass divided by however long it took the time. And that gives you then your change in mass per day, per hour, whatever it may be. So that's all of B4. Next topic is B5, which was all about genetics. So the first thing we need to understand is what is variation, because we're going to use this term a few times. And quite simply, variation is just the differences within a species. And generally, when we're talking about variation, we're talking about what we call the phenotypes. And a phenotype is an observable characteristic of the organism. So that's things like blue eyes, brown hair, anything that you can just look at and go, it's this, then that's your phenotype. Now, what we find is that with any species, then we do have this variation, these differences, and we can actually categorize them into one of two types. It's either going to be genetic variation or environmental variation. So genetic variation, as the name suggests, is caused by the genes inherited from the parents. So this is things that you can't change, really. So it's things like your blood type, your eye color, they're fixed by your genes. You're born with them, and that's your lot. The environmental variation is caused by the environment in which you live. So this is things like your accent, whether you've got scars, tattoos, piercings. All of those are things that you've actually got as a result of your existence. You weren't born with them, and I'm sure many babies are pretty pleased that they're not going to be born with their parents' tattoos and piercings, but you can choose to have those in your life. The one thing to bear in mind here, though, is it's not just the case of every particular characteristic is caused either by genetics or by the environment. There are some that are both genes and the environment that come into play. So things like your height, for example, yes, your genetics play a part in that, because if you've only got parents who are 5 foot tall, you're not going to be 6 foot something in all likelihood. But same reasoning here, if you've got 2 6 foot tall parents, then if you don't get a good diet when you're growing, you're not going to get to that possible maximum height either. So we've got both environment and genetics come into play. When we're looking at variation, then, it can actually be categorized as either continuous or discontinuous. So continuous variation is the one that could take any value within a range. And this is the kind of variation that's caused by both genetics and environment. Now, what we usually find here is that if it's genetic, then it's controlled by multiple genes. So this would be things like your height, or it could be the length of fur in animals, for example. It can take any value within a given range. Now, when they ask you to plot a graph of this, or to suggest what type of graph we should use for continuous variation, then you need to say it's a histogram. And the histograms are the ones where there's no gaps between the bars. If we talk about discontinuous variation, then, these are the ones that can only result in a discrete value. And these are caused by genetic variation, which are controlled by only one or a very small number of genes. So it's things like your gender or your eye color. You are one or another. So it falls into very clear categories. And as a result of that, we display that on a bar chart, which is the one that does have gaps between the bars. So just go careful on that one. When we're considering actual reproduction, then one of the types of reproduction we see is asexual reproduction. Now, asexual reproduction results in a clone, and a clone is an organism that is genetically identical to its parent. So a good example of organisms that produce asexually is bacteria. So hopefully we remember that bacteria are going to reproduce using that process of mitosis, whereby we get the DNA replication, the chromosomes line up along the equator of the cell, pull to the opposite poles, and the cell divides to make 2 identical copies of that parent cell. Now, good examples of the plants that will do this are things like strawberry plants, potatoes, daffodils. Any of those are going to produce these identical clones as a result of asexual reproduction. It's not just plants, though. We do have a few animals that also can reproduce asexually. So things like starfish and sea anemones, they do use asexual reproduction as well. The other option, and the one that's more common in animals, is sexual reproduction. Now, for sexual reproduction, we need 2 parents. So what we're going to see as a result of this, because we're getting DNA from both parents, then we've got more genetic variation occurring. So what we're going to see as a result of this are quite significant differences between the individuals in the species. When we talk about sexual reproduction, then we are needing gametes, which are the sex cells. So if we think about humans, that's the sperm and the egg cell. Now, eggs are made in the ovaries in the female, sperm cells made in the testes of the male. and they're made through a process called meiosis, meiosis. Go careful here. When you are looking at meiosis and mitosis, then the difference between you getting a mark and not getting it is about your spelling. Now, we're not overly strict on it. You don't have to have it word perfect, but it's generally a case of we're looking for that T. So mitosis must have the T in it. Meiosis must not have a T anywhere near it. And I've seen a lot of random words where people have merged the two together to make some process that just doesn't exist. So sexual reproduction, meiosis, asexual, mitosis. Now, when we're using meiosis in our sexual reproduction, we produce these gametes, these sex cells. And the key thing about them is they are haploid, which means they've only got half the number of chromosomes of a normal body cell. Do go careful here, because obviously we know in humans a normal body cell has 46 chromosomes, and therefore our sex cells, the sperm and the egg, will have 23 chromosomes. They don't always ask you about humans. Sometimes they will put it into a random context of a creature you know nothing about. They might decide to tell you about polar bears, for example. In the question, they will tell you either how many chromosomes are present in a normal body cell or in the gamete, the sex cell. And then they could ask you how many chromosomes are present in the other one. So if they've told you that a polar bear has 14 chromosomes in a normal body cell, and they ask you how many chromosomes are present in the sperm cell, divided by 2, you've got it. If they tell you how many chromosomes are present in the sex cell and ask you how many are in a normal body cell, times it by 2, you've got your number there. Just make sure you read the question to get that number and don't just assume it's 46 and 23, because that is just us as humans. Now, what we find then is, as a result of having these two gametes, during the process of fertilization, they join together, and therefore we have the full complement of DNA we need for our organism to produce the offspring. For those of you doing GCSE biology, you do need to know about comparing asexual and sexual reproduction. So kind of like the pros and cons lists of using them. If we think about asexual reproduction, first of all, a couple of key advantages. First of all, the parent, if they're well adapted to the environment, then their offspring will be too, because they're genetically identical. So they will be the same as their parent, just as well adapted. And we only need one parent, so reproduction is much faster. The downside to asexual reproduction, however, is that if we see a change in any biotic or abiotic factor, it can wipe out the whole species. Because if they've all descended from just that one parent and they're all genetically identical, something that one of them is susceptible to, they all are. The difference with our sexual reproduction then, key positive about sexual reproduction is variation. Anytime we get variation, then we have a chance of adapting to the environment better. A downside, though, is that we need two parents, therefore reproduction is going to be a slower process. And bear in mind that certain organisms have kind of got around this to get the best of both worlds, and they use both sexual and asexual reproduction. So things like a sea anemone, for example, uses a bit of both. Now, what we actually then come on to is a second phrase that we haven't mentioned, which is diploid. So diploid is just where we've got two copies of the chromosome. So haploid, one of each chromosome, diploid, two of each chromosome. And again, that's just where they could tie in that question looking at how many chromosomes are present in a haploid or a diploid cell. So just bear that one in mind. If we're talking about sexual reproduction, there is one exception to the fact that we get different individuals as a result of sexual reproduction, which is identical twins, obviously. So just bear that one in the back of your mind, just in case. Now, the next process is good old meiosis. If you've already learned mitosis, the good news is that you already know half of meiosis. So that's good news. Now, meiosis makes the gametes, the sex cells, as we've said, and from our original parent cell, we are going to produce 4 haploid cells, so 4 of our sex cells. Now, the location of this in humans will obviously be the ovaries and the testes. In terms of how meiosis takes place, first thing that happens is the chromosomes are copied, so we get the DNA replication. They then line up along the equator in their pairs, and then they're pulled to the opposite poles, one of each pair of chromosomes going to each end. The cell is then going to divide in that process of cytokinesis. So hopefully at this point you're sitting there thinking it's just the same as mitosis, and it is. So DNA replication, line up on the equator, pull to the opposite poles, cell divides. That's our first bit. Then what we see in meiosis is the fact that the chromosomes then line up along the equator again in our two cells that we've got at this point. Those chromosomes are pulled in half, so half go to the top, half to the bottom, so to the opposite poles again, and the cell divides into two. So that means we've now got 4 cells, each with half the number of chromosomes present. So that gives us our 4 gametes. All we need to remember is DNA replicates, chromosomes line up on the equator, pull to the opposite poles, cell divides, line up on the equator, pull to the opposite pole, cell divides. That's it. And then you've got the whole of meiosis there. What we actually find is that we get variation in this sexual reproduction as a result of 3 things. First one is the fact that during that point where they're all lined up as the chromosomes along the equator, then we get something called random assortment. It's not a case of all of the chromosomes that originated in the father line up on one side and all of the ones that originated in a mother line up on the other. It's completely random how they do that. So that means that as they're pulled to the opposite poles, you're going to get a different mixture in each of those cells. We then also have a process called crossing over, which is where little bits of the chromosomes get swapped amongst each other. And then finally, once you've actually got the sperm and the egg, then when they come together, it's completely random as to which sperm will fertilize which egg. So again, we've got those 3 stages that lead to the variation. Within the actual DNA, then what we've got are genes. Hopefully you remember that from earlier on. So your section of DNA can be divided up into small little regions called a gene, and a gene controls a certain characteristic. Now, those genes actually have different forms called alleles. So you've got a gene, for example, for eye color, but the alleles would be blue eyes, brown eyes, etc. So the allele is just the different form of the gene. Now, what we'll actually find is, depending on what alleles you inherit, that's going to determine your characteristics. So, some more terms we need to know for our genetics. Dominant alleles, these are the ones that are the strongest, if you like, okay? So what that tells us is if you have 1 dominant allele or 2 dominant alleles for a particular gene, then that trait will be shown. And we represent that in any diagrams with a capital letter. If we're talking about the recessive alleles, then you will only express that characteristic if you've got both recessive alleles. And we use a lowercase letter to show those in our genetic diagrams. Now, what we're actually looking at here is something called the genotype. So what we're looking at is the combination of alleles that are present within an organism. You may be one of 3 things for these genotypes. You could be homozygous dominant, which is where you've got both alleles are dominant. You might be homozygous recessive, both alleles are recessive alleles. Or you could be heterozygous, which means you've got 1 dominant and 1 recessive. So homo, same, hetero, different. And the zygous is just about the alleles there. Now, what they could ask you to do is to actually draw a genetic cross diagram or a Punnett square to work out the likely offspring of 2 particular parents. Now, there's a few stages that you go through to get this right each time. First thing is you state the phenotype of the parent, so brown hair, blonde hair, whatever it may be. You then state the genotype of the parent, so the capitals and the lower cases. You state the gametes, so you split those into the individuals. You draw your Punnett square, which is the one that looks kind of like a window. So you set out your little Punnett square like this, and then you're going to work out what fills in, and from that you then state the phenotype for the offspring as well. Now, if we have a little look at an example, what we're going to have a look at is inheritance of hair colour in this case. So what I've just drawn for you there is your outline, okay? So we've got 2 brown-haired parents because brown is going to be our dominant trait. So they are going to be heterozygous, so 1 dominant allele, 1 recessive allele. So both parents have brown hair because there's 1 dominant allele for each. And then all we're going to do is whatever is up at the top here, we fill in to the 2 boxes beneath. So we start off by putting our capital B in there. Do the same for this one. It's a lowercase b, so we put the lowercase b in for those ones. And then we do the same thing going across. So in this case, we end up with those as our potential genotypes of the offspring. We then need to identify what their phenotype will be, so what colour hair they've got. Now, any with a capital B will have brown hair because that is the dominant trait. And any with only the lowercase b are going to be the blonde-haired ones. So you just write in there that these three are going to have brown hair because they've all got a capital in there. This one is going to be the only one with blonde hair because they're both recessive. And then the last thing you need to do is write down the percentage. So in this case, there are 75% that will be brown haired. You could also write that as 3 out of 4. So it's up to you which way you write it, but you do need to put that percentage in at the end. The other one they could ask you is about the actual sex determination. So whether you're male or female. And that's determined by the X and Y chromosomes. So the key thing to remember here is that males are an X and a Y, and females are 2 X's. So just make sure that when you're writing about this, if they ask you what chromosomes are present in a male, then you need to write X and Y. Don't just write Y because you've just forgotten half of their chromosome at that point. Okay, so X and Y for male, X and X for female. So you've got to have the 2 X's. You can't just write X and that's it. Now, what we find is that once you've actually got your DNA, that's not it, okay? Because we also throughout our life may experience what's called a mutation, which is a change in the base sequence of the DNA. And these mutations can occur spontaneously during DNA replication. But there are certain things that will increase the chance of this mutation occurring. And 2 of the common ones are ionizing radiation like ultraviolet or chemicals, things like benzene, for example. Now, what we then find is that as a result of this mutation, we get genetic variation because we're changing alleles. Now, these mutations may show no effect on the phenotype at all, so you don't even realise anything's happened. They may influence the phenotype, or they could actually completely determine the phenotype, which then leads to that variation in the species. And when we're looking at these changes that could take place as a result of a mutation, they're either going to be neutral, doesn't help the organism or hinder it. They could be harmful, therefore creating problems for the organism, or they could be beneficial, making it better for the organism in some way. So a neutral mutation would be the ability to roll your tongue. So it's not affecting your ability to survive if you can roll your tongue or not. It's just something you can do. Something that would be harmful would be cancer, because that's always a good mutation there. That's a really nice example of how harmful it can be. We could also have cystic fibrosis in there, which is a change in the channel proteins. Or we could also have sickle cell anemia, which is a change in the protein molecules. In terms of beneficial, so the ones that are actually advantageous to have as a mutation, then things like antibiotic resistance in the bacteria or skin colour is another advantageous mutation. If you're doing GCSE biology higher tier, you need to know a little bit more about these mutations. So your DNA is actually organised into coding sections, which are then separated from each other by these non-coding sections of DNA. Now, when we get this mutation taking place within a gene, we could see one of 3 things happening. We could either change a DNA base, so just swap it for a different one. We could add a base into it, or we could delete a base from it. If we just change a single base, then that's going to change just 1 amino acid, because hopefully you remember from our earlier work in B1 that we're going to read that strand of mRNA in a sequence of 3 bases at a time, a triplet. So each triplet codes for 1 amino acid. So if we change a single base, we've changed a single amino acid. If we add a base into the sequence, then that's going to have much greater effects, because it's not only going to change the amino acid where it's been added, but everyone after it. Because if you stick an extra letter in, you've shifted everything along one place. So you've changed all of the triplets coming after that. And it's the same effect if you delete a base, then all of those triplets that come after that point could also be changed. So just bear that one in mind in terms of the extent of them. Still, just for higher tier biology folks, you also need to think about the fact that if we change the amino acid, then we're changing the protein. because the sequence of the amino acids determines how that protein folds up, and therefore we could change the shape of it, particularly problematic if we're talking enzymes with their active sites, or we could make completely the wrong protein. So do remember that link back to the work we've done on enzymes, transcription and translation in B1 there, because they do say that they could pull on your prior knowledge in this paper too. What we also find in terms of our DNA is that we've got this little codon called a start codon. Now, that's before each gene in the non-coding section of the DNA, and that tells it this is where you start transcription. If the mutation occurs in the start codon, then we may not transcribe the gene at all, which means we won't make the protein. So bear in mind the start codon is also an important place there. Still, just for the GCSE biology higher tier folks, you need to know about the history of genetics. And the most important person here is Gregor Mendel, the father of genetics. And he spent a lot of time in the gardens playing with pea plants. And what he actually did was he observed that characteristics were passed on from parents to the offspring. And those characteristics in plants, he said, were determined by hereditary units. And those units are passed on, one from each parent, and they can either be dominant or recessive. So this is where we started to get the idea of genetics. Our second scientist is Friedrich Miescher, and he discovers nuclein. And that's just this acidic substance present in the nucleus. Third one is Oswald Avery, and he actually transferred DNA between bacteria. So he showed that these genes, therefore, are made of DNA. Next one is Erwin Chargaff, and he discovered base pairs. So he's quite an important guy in the grand scheme of things, because he found that even though different organisms have different amounts of DNA, all of that DNA has equal quantities of adenine to thymine and cytosine to guanine. So he then said, if we've got the same amount of adenine and thymine, they must therefore pair together. And the same with cytosine and guanine. So he came up with those base pairs. Next up is Rosalind Franklin and Maurice Wilkins. And Rosalind Franklin was the one who did the X-ray photographs of the DNA crystals themselves. And it was from that X-ray photograph that she took that then finally James Watson and Francis Crick were able to identify the double helix structure of DNA that we know today. And do remember that double helix structure of DNA is held together by those complementary base pairs, A's with T's, C's with G's. Since then, which was all the way back in the 1950s that we discovered this, we didn't just think, we're done, again, that's it, we don't need to know anything else about genetics. Since then, what we've actually gone on to do is identify individual genes that code for certain things. So we've also developed the field of genetic engineering as well. Back in 2003, we completed the thing called the Human Genome Project, which is where we've now sequenced all 24,000 genes within the human body. And since then, what we've been doing is working on ways that we could replace faulty genes with normal copies through that process of gene therapy. Obviously, you do need to try to remember those scientists in the correct order, so try and come up with a mnemonic that works for you for that. Back for everyone, so combined, biology, foundation, higher, whole lot of you again, where we're coming on to look at natural selection. Now, what we're going to have a look at here is the process of natural selection, which then leads to evolution. And evolution is the gradual change in a species over time. And the way they do that is through this process that was suggested back many years ago by good old Charles Darwin. Now, natural selection is how we get evolution. And what we've got are 5 key stages to this. Step 1 is going to be variation, which means that in our species, as we've already said, organisms show variation. And as mutations occur, then variation will increase. Step 2, survival of the fittest. The organisms that are best adapted to the environment are those that will survive. Those that aren't well adapted, they're going to die. Third step is reproduction. So those that have actually survived longer are therefore more likely to reproduce. Fourth step, and therefore they're going to pass on that advantage because the genes are obviously going to go from the parent to the offspring, which means those that were more likely to survive therefore have reproduced and passed on those advantageous genes to their offspring. And this is then going to take place in the 5th step over many, many generations, which over time could lead to the development of a new species. So remember those 5 steps. If ever they ask you to explain the process of natural selection, variation caused by mutations, the best adapted to their environment survive, they're going to reproduce, passing their genes on to the offspring, and it repeats over many, many generations. And that's it. So even if it's a 6-mark question, you've covered the whole lot by doing that. Good examples of natural selection that we've got, the peppered moth, first of all, which is obviously we used to have these 2 different colours of moths that were very common. We had the sort of light coloured ones and the dark coloured ones. And before the 19th century, the majority of these peppered moths were actually light in colour because they camouflaged beautifully on the trees, and the dark ones stood out and got eaten. After the Industrial Revolution, then we had a whole load of pollution and soot covering everything, rather dirty place to live. And therefore, those lighter moths stood out and the birds ate them. The dark moths were better camouflaged, they survived. And so we saw the change from lighter moths being more prevalent to the darker moths. So it was natural selection. They were being selected because they were better adapted to their environment. Another good example is antibiotic resistance in bacteria, that obviously those that do have the resistance to antibiotics are likely to survive. If we then come on to think about evolution, then this is widely accepted these days. There are still some people who don't, but we won't worry about them today. On the most part, people accept evolution because we've got really good evidence for it. So the first bit of evidence is all about fossils. Now, a fossil is formed when the remains of animals or plants get mineralized, and when we're looking at the fossils and we're digging them up underground, the deeper we go, then the older the fossils will be. So near the top, we've got young fossils, further down, older fossils. And if we actually dig up all these fossils from the different layers of rock, then we can produce a little sequence to show how the organisms gradually change over this long period of time. And that's what we have with the fossil record, is this gradual change in a species over a long period of time with physical evidence of those changes taking place. What we also find from looking at these fossils is that the oldest ones, so if we did go back to our oldest rocks, then they contain fossilized bacteria, whereas more recent rocks have more complex life forms. So this does actually suggest the idea that life on Earth actually started off as bacteria and then became more complex as the years went by. We will also find the fact that plant fossils occurred before animal fossils, which tells us that before animals could actually evolve, we needed plants to evolve. So we can compile a lot of information from that. We also get information about who's related to who from the fossils. So we can actually see that we've got closely related organisms that actually came from the same ancestor in their family tree. In terms of other evidence that we've got for evolution, we can use things that are very quick at reproducing, things like the bacteria, for example, because we can see many generations in actually a short space of time. So we can see that evolution taking place in our lifetimes. And any organism that reproduces quickly is good for that. So it's not just bacteria, there are certain types of fish as well that work there. Other bits of evidence we can use is the comparison of things like DNA and also the proteins. So we can do what's called a molecular comparison of these biological molecules, because closely related species have the most similar DNA and the most similar amino acid order in their proteins. So by looking at the molecular comparisons between the DNA and the proteins of different organisms, we can work out who's most closely related. Obviously, not everything is going to evolve. What we do find is that when we get these changes in the environment, then some things aren't adapted and they die. And what we actually have is a situation now where 99% of all species that have ever lived on Earth are now extinct. For those of you doing GCSE biology, you need to know a little bit more about the theory of evolution. Now, what we actually have is pre-Charles Darwin, then the accepted theory of how all species on planet Earth were created was God did it. Now, obviously back in 1831, then Charles Darwin went off on his wonderful voyage on HMS Beagle and travelled around, okay? The most famous bit that he travelled around were the Galapagos Islands. Now, at the time that he was travelling, then he'd also read a book on geology by a guy called Lyell. And Lyell had suggested that fossils were evidence of animals that lived millions of years ago. So Darwin had that in his head already, that we've got some evidence of these animals that lived a long time ago through these fossils that we found. And as he was traveling around, he took these very detailed documents, sort of readings and drawings and actual samples of these organisms he came across. And one of the most famous ones are the finches. Now, when he was looking at these different finches on the islands in the Galapagos, then he looked at the shape of their beaks and what food sources were present. And from that, he then eventually came up with his theory of natural selection. But this is where we sort of have to diverge a little bit, because at the same time that Darwin was doing this, then in Borneo, which is another country in the world, then we actually had a Welsh scientist, which is Alfred Russel Wallace, who was actually doing a rather similar thing. And he was actually looking at this and came up with the same idea, that these organisms were actually going through this process of natural selection and evolution. And what Wallace did was he sent his ideas to Darwin for peer review before he actually published. And because they were so similar, they actually presented their two papers jointly. Now, peer review, if you remember, is where a scientist will send their work to another scientist so they can read through it, check for accuracy, and obviously pick up any problems and so forth. Obviously, when this was all then put out into the general population that was still sitting there going, things live on Earth because God made them, that was it, then there was a bit of kickback. People didn't just go, Charles Darwin said this, we're good. Instead, he was quite mocked because his ideas quite clearly went against the Bible. If we're no longer sitting there saying God created all species, but actually they had this process of natural selection and evolved over time, people didn't like that. And so you got things like the old newspaper picture of Charles Darwin's face stuck on a monkey's body, because that was the whole thing, that, you know, we came from monkeys. No, we didn't. We evolved from a common ancestor that's shared with monkeys. Obviously, lots of mocking at the time. Today, the vast majority of people do accept this because we've got all of that evidence we've already discussed. Combined Science Biology Foundation hire everyone back again to look at our classification systems. Now, classification is sorting the living organisms into groups based on similar characteristics. And what we can actually do here is, by classifying them, it can help us to identify species, it can help us predict characteristics and show these evolutionary links between them all. When we talk about a species, we're talking about a group of organisms which are able to reproduce and produce fertile offspring. Used to be a 2-mark question. So the key thing there is reproduce together, first mark, producing fertile offspring, second mark. And what we actually have here are 7 taxonomic levels. So kingdom, phylum, class, order, family, genus, species. Again, come up with a helpful little mnemonic to help you remember it. I have one that I use with my 6th formers. I'm not going to share with you here because it's not the most appropriate for you when you're still at sort of GCSE level. But come up with one that works and sticks in your head to get the correct order there. Once we've actually got that in our heads, then we've got 5 kingdoms that every living thing on planet Earth can be actually divided into. So the 5 kingdoms are plants, animals, fungi, Protoctista, and the prokaryotes. And the way we're actually going to divide them up is mainly by using one of 2 systems. We'll either use the artificial classification system, which is the more common one, where we're looking at the observable characteristics. So we'd be looking, do they have wings? How many legs have they got? Things like this, the bits you can look at and go, well, this one's got 4 legs, that group. This one's only got 2, that group. So they're the observed ones for the artificial classification system. Downside to that is it doesn't consider evolutionary relationships at all. The other type of classification system we can use is the natural classification system. And this one is based around these evolutionary relationships, and it's determined through DNA analysis. So what we're doing here is something called phylogeny, which is the study of the evolutionary links through the study of the DNA itself. And from that, we can build up a family tree showing how all of the organisms are actually related to each other and work out which ones are most closely related. So that's what we're looking at in terms of our classification. That's all of B5. So last one is B6, which is the chunky monkey of all of these. So the first part of this is looking at sampling techniques, which is also one of those assessed practical tasks. Now, in terms of our sampling techniques, there are a range of different things that we can use here. Hopefully you've all used these and therefore know what I'm talking about. So we've got a quadrat, which is basically that big metal square of a fixed area, and we're going to use that to sample plants. You don't use it to sample animals because animals move, and the quadrat doesn't. So literally, the quadrat is a square of a fixed area that we're then going to randomly place on the ground, and we're going to count what plants are in there. Pooters are the little things that you're going to be sucking through one straw and sucking a little bug in with the other one, making sure obviously you suck through the straw that's got a little mesh cover on it. Otherwise, when you've collected a few bugs and then you suck through the wrong straw, mouthful of bugs, not pleasant. So pooters collect small invertebrates. Your pitfall traps are where we bury a jar underground so that the top of it's level with the actual ground itself. Put a little roof over it so it doesn't fill with water, and then you just leave it, go back and collect what's ever walked in and fallen in overnight. We've got sweep nets, which as the name suggests is a big net, which you sweep through long grass in order to pick up things like grasshoppers. We've got transect lines, which we're going to talk about a little bit later on, but basically it's a tape measure we're going to run along an environment and then take samples at fixed points. We do kick sampling, which is in water, where, as the name suggests, you kick the edge, and that dislodges the creatures and the things in the riverbank, and then you collect them with a net downstream. And tree beating, you stretch a big white sheet under a tree, and you give it a shake and see what falls out. So those are the different things we can use to carry out sampling. Once we've actually gathered living things of some description, we need to know what living things they are. And we're going to use things called a key to do this. Two types of key we can use: the spider or branched key, first of all, which starts off with your question, and then you've got two options that take you to the next. So we might have our plant at the top, and then you've got two options: broad leaves here or narrow leaves there. So if it's got broad leaves, you go this way, and then you do the same, just dividing further and further down until you're left with just one species. The other one, the numbered key, this is where you've just got a list of questions, kind of like those choose your own adventure stories that you probably read as a kid. So you kind of read what the question is, and then it tells you go to question such and such, and you just keep doing that until you find whatever it is at the end. One of the techniques you need to know about is the capture-recapture technique. So this is what we would use for things like grasshoppers and bits like this if we want to know a total population size. So what you do is you go out and you take your first sample. So you capture them, you count them, and you're going to mark them. The key thing about marking is you must do it in such a way that it's not going to adversely affect their chance of survival. So if we're talking about grasshoppers, you're not going to paint their back bright red, because then they're going to stand out and birds are going to eat more of those ones. So you need to mark them in a way that you can see, but doesn't mean they're all going to die. Then what you do is you release them and go away for a fixed period of time. At a later stage, you then come back, use the same technique and the same equipment to recapture another sample. You count them, including the number that are marked and the number that are unmarked. Once you've got that, what we can do to work out the population size is the number in the first sample times the number in the second sample, divided by how many in that second sample were already marked. And that then gives you an estimate of the total population in the area. Two assumptions we are making here though. First of all, nothing died. And secondly, we didn't have any immigration or emigration. So none of them left or joined the area. So a couple of key assumptions we make at that point. Whenever we're talking about sampling, then we need to think about how we're going to do this. Not just in terms of what equipment we're going to use, but also in terms of the process. And what we want to do in order to get the most accurate estimate of a population size is use something called random sampling. Now, quite often the way we do random sampling, if we're looking at the school field and quadrats, for example, is we'd use two tape measures to draw up a grid. So you'd have one tape measure going on one side and one on the other side to form like axes. What we then do is use a random number generator to give us coordinates. So we get two numbers, which would then tell us if we had like a 2 and a 3, you go 2 meters along your x-axis, 3 meters up the y, and that's where the quadrat goes. And the whole idea of doing this random technique is to remove any bias. Because I know, and you probably remember this from where you did it, that when your teacher took you out to do the sampling, if they didn't make you do this, then when you had your quadrat, you kind of wandered out, you had a little look around, found where there was something that wasn't just grass, and put your quadrat there to count something that was a little bit more exciting. So that's obviously got a lot of bias in there and is going to lead to quite a skewed population estimate. So we need to use the random sampling to remove that bias from it. There are instances where we don't want to use random sampling. So if we want to see how the distribution of organisms changes over a certain area, it might be sort of going up a beach, for example, further from the sea. It might be coming out from under a tree to increasing amounts of light intensity. But if we're studying how that distribution changes over a distance, we use non-random sampling. And we do this with a transect line. So you run a tape measure out across the area you wish to sample, and then you're going to place your quadrat at fixed intervals along that line. So every meter, every 2 meters, whatever it may be, and then you record what organisms are present and how many of each there are. That then allows you to plot the thing called a kite diagram later on, which just has your central line, and then you would have bits sticking off based on how many there are. So it will look something like this. So the actual width here tells you the number that there are. So we would have obviously the distribution changing the further away you got from whatever it was, whether it's the sea, the tree, whatever. The next thing we need to come on to then is biodiversity. Now, biodiversity, quite simply, is the variety of living things in an area. And it's essential for maintaining this balanced ecosystem. Even us as humans rely on this biodiversity for a whole range of raw materials that we need in our lives. But we as humans are also the leading cause of the loss of biodiversity, all down to our ever-increasing human population. So one of the key things that we do here is deforestation. So that's the permanent removal of large areas of forest. So we'll be taking out trees to build things for fuel, just to create space so we can put more buildings in. And as a result of that, we've reduced the number of trees, and therefore we've reduced the number of animal species that would normally rely on those trees as well. We've also got the problem of agriculture. As obviously the population increases, we need to feed more people, so we need more farming land. So we're going to be using more intensive farming techniques to actually produce enough food to feed everyone in the world. As a result of that, we're using a lot of chemicals. So things like pesticides could actually remove food sources for organisms. We could also see things like bioaccumulation, which is where some of these pesticides don't break down easily. So we might use it to kill a little bug, but it builds up as other animals eat those little bugs until it can actually poison the larger creatures as well. Things like herbicides, which are used to kill weeds, will also reduce the number of plant species present and therefore reduce what can live in that particular area. If we're thinking about other human actions, we've got hunting and fishing. Obviously, that's going to reduce the population there. Things like overfishing can actually eliminate an entire species from a particular region. So as a result of that, we could lose them from that place. If we're hunting things, whether it be for food or just because you're one of those people that kills animals for kicks, then we could decrease the food for another organism or actually take a key part of a food chain out. So it can have much wider impacts. And the last one we've got there is pollution. So the more polluted an area actually is, the fewer organisms that can survive there. And a really good example of this pollution is eutrophication. So when we're using fertilizers in order to grow more and more food in that limited area, then the fertilizers run off the fields into rivers and lakes. That results in an increased amount of nitrogen within the water. We get an algal bloom, which is where the algae population just explodes, and that's going to block the light. So any of the plants in our lake or river will die. As they're dead, then decomposers are going to break them down, and through that action, they deplete the level of oxygen within the water, and it kills the fish. So just by spraying too much fertilizer on a nearby field, we've now killed all of the fish in a lake or river. So eutrophication, make sure you know those steps there. If we're thinking about species, there's a couple of terms that we've heard bandied around quite a lot, I'm sure. Endangered species, where we've only got a very low number left of them, and therefore they're at risk. And then extinct, we've killed the whole lot. So there's no more left in the world, they're just gone. Now, what we're going to try and do in order to help the endangered species and prevent them becoming the next extinct one is the process of conservation. Now, conservation is all about protecting the natural environment to ensure habitats aren't lost. And we're using a process called active management of our habitats. So that just tells us we can still use resources, but we're doing so in a sustainable way. So it's the idea of, if you cut down a tree, you plant a couple in their place. So even though you're using a resource, you're not taking it away permanently, because they will regrow. Other things we can do is control grazing of an area, so not letting the entire thing get destroyed. We can restrict human access, or we can go along the route of reintroducing species to an area where they may no longer be. One of the key things about reintroduction is we need to have some species to reintroduce, and we use captive breeding programs for this. So this is one of the main parts of zoos. It's not about just going to a zoo and looking at the pretty animals. It's actually that a lot of zoos have captive breeding programs to increase the numbers, so they can then be reintroduced to the wild at a later stage. And the whole idea behind this is to create that stable and healthy population of a species that can be gradually reintroduced to their natural habitat over time. And I know down here in Kent, then we've got places like Portland and Howlett that do really well with the gorillas, for example. The downside to this, though, is we won't necessarily have vast numbers of these organisms in captivity. So maintaining the genetic diversity can be quite difficult due to limited numbers of breeding partners. And we might also breed organisms in captivity that aren't suitable for release. So it may be a case of they don't have the ability to go back into the wild because the actual area they used to live in isn't there anymore. Or it might be that one of their food supplies isn't there, so we can't then release them. If we're talking about plants, then it's not just a case of animals we're worried about. We do worry about plants as well. And in order to prevent the extinction of plant species, we've got seed banks. So these are actually these big vaults, basically, that are generally in very, very cold places. And we have then got these massive stores of seeds of any kind of plant you can imagine. The whole idea being that should that plant become extinct as a result of something that happens in the environment, we've got the seeds that we can then regrow them in the future. In terms of trying to maintain this biodiversity then, what we need is not just one person trying to work on this or one little area. It's got to be looking at local and international cooperation in order to do this properly. So what we've got are things like international organisations which are trying to secure these agreements between all the nations of the world. So you've got things like the IUCN, which publishes the thing called the Red List, and that's all about informing people about which species need conservation. So they're rated on that scale, and we can see which ones are endangered, which ones are critically endangered and need urgent action. We've also got the Convention on International Trade in Endangered Species of Wild Flora and Fauna, and that one is all about regulating all of the trade, as the name suggests, of any of these wild plants and animals and their products. So looking at trying to stop people trading in ivory and all of those things, which then means it's less attractive to kill those organisms. We've also got the Rio Conventions, which require countries to develop strategies for this sustainable development, reduce their greenhouse emissions, and combat desertification, with the whole overall goal here of maintaining biodiversity. Now, reducing greenhouse emissions is obviously a key one in a lot of countries, exception being America, obviously with good old Donald Trump, but most countries are actually working towards getting their carbon dioxide emissions down quite considerably through various aspects, whether it be getting rid of fossil fuel use or changing certain aspects of their everyday practice to bring that amount down. Now, in terms of the local agreements, this is where different countries are going to be focusing on different things and using different schemes to achieve this biodiversity maintenance. So in England, we have things like the stewardship schemes, where we offer farmers payments to conserve the landscape. So rather than just sort of letting farmers wipe it out and just use monoculture with everything in that entire area, then they will be offered a payment to not do that in a field, just to let it go wild and have the natural flowers and plants growing there. If you're thinking about what you as an individual can do, then we can also look at things like tourism. Now, tourism is big business for a lot of countries. In fact, it is their key business for some of them. It brings in that money, and if they're good, they can use this money to then extend and improve habitats. They can use it to employ people like game wardens in order to prevent poaching. We can use it to put things into place to maintain this biodiversity. And ecotourism is all about ensuring that the tourism that you've got in your country doesn't have a negative impact on the natural environment or the local communities that live there. So it's just looking at things like keeping tourists on footpaths, keeping them out of certain areas. So if you go to a country where, for example, you've got sea turtles that are coming to lay their eggs on the beach, then keeping those beaches as non-sunbathing beaches, because obviously the eggs are buried under the sand. You don't want people trampling around on the surface there. There are some negative impacts though, because if we're telling everyone they must walk along a certain path, then what we can see is contribution to things like soil erosion. So it's not perfect. Now, for those of you doing GCSE biology higher tier, you need to know about how we can monitor this biodiversity in any of our schemes and whether it's actually having any impact, full stop. So what we're going to do is take regular samples of plants and animals in order to monitor the type and number of them present. And one of the key things we're looking for here are things called indicator species. So these are just organisms that we can use to measure the environmental quality, either because they're present or they're absent. So that's what our indicator species is. If we think about air pollution, first of all, then sulfur dioxide is one of the main sort of forms of air pollution, one of the more common ones that we have. And we get it from the combustion of fossil fuels. Sulfur dioxide is bad because it's going to lead to acid rain, and acid rain can kill trees, fish, etc. One of the key indicator species for air pollution is a substance called a lichen. Now, lichens don't have a root system. All of their nutrition comes from the air, so they're super susceptible to air pollution. Now, we do have some that are really good at coping with pollution, and they're the ones that you're going to find in the industrial areas. They generally look like the gross, crusty ones. And other species of lichen will only grow in areas where the air is beautifully clean, and they tend to be the lovely branched, flowery looking things. So you can just look and see what lichens are present, and it gives you an idea of the air pollution in the area. Water pollution is our next one, and again, this is just caused by releasing harmful substances into lakes, rivers, seas, etc. The higher the level of pollution, then the lower the dissolved oxygen levels in the water will be. So what we can use are indicator species that tell us how much dissolved oxygen is present, and that then tells us how much pollution there is in turn. So if we've got unpolluted water, you're likely to see things called mayfly larvae. If it's low, it'll be freshwater shrimp. High, we're looking at water lice, and very high, the good old sludge worm. As the name suggests, loves the sludgy, gross water. If you're doing GCSE biology, but foundation and higher, you need to know about food security, which is the ability of the human population to access affordable food of sufficient quality and quantity. So we've got a few factors that are going to actually impact our ability to do this. First of all, the increasing population of humans again. It's a recurring theme in this paper, as you can tell. Secondly, the changing diets. So we're now eating a more varied diet with a lot more meat products in there. Third one, climate change, more droughts, expanding deserts, etc. And the last one we're getting more pathogens, so new pathogens, new pests evolving. Now, we can take certain steps in order to try to combat some of these things and therefore meet the demands of our population. Things like taking steps to maximize photosynthesis, so using greenhouses or glass houses, as OCR used to refer to them as, that can then allow us to control the light intensity, the temperature of the water, everything to maximize photosynthesis. We can use chemicals like fertilizers to replace any lost minerals. We can remove competition from pests and so on. We can also use pest-resistant varieties of crops rather than just taking a gamble with wild varieties. So any of those things are going to contribute to giving us more food, which then allows us to feed more people. 4 key chemicals that we could use here: fungicides to kill any fungi, pesticides kill any pests, insecticides kill any insects, and herbicides don't kill herbs, they kill weeds. So side kills, thing in front, what it kills. We got 2 methods of producing food. You've got intensive farming or organic farming. Now, intensive farming is the one that uses the techniques to produce the maximum yield of food from the minimum area of land. So this is the one that's using any chemical imaginable: fertilizers, pesticides, herbicides, all of them. We're going to be using techniques to maximize animal growth rates. So rather than having them free roaming in fields, we keep them rather restricted in their movements. So when we've got all the chickens packed really quite closely together, for example, we're using a lot more machinery rather than humans in order to minimize labor. If we're talking organic farming, pretty much the opposite. We're using natural methods here. So we're not using chemicals as opposed to artificial fertilizers we're using horse manure. Rather than using a lot of machinery, we're using a lot more manpower on the ground. We're actually allowing the animals to be free roaming with their free range eggs and so forth. So that's organic, intensive, chemical it up, lots of equipment, and basically maximize what we can get from every given meter squared. If we talk about sustainable food production, still just for GCSE biology folks, then this is producing food in ways that could be continued indefinitely. And this is obviously the goal of life. We want to be able to produce food to feed the population for the future. Okay, we don't want it to be, well, for the next 10 years we're good, but after that, who knows? We want it to be sustainable. We want to be able to do this indefinitely. And this is where we're going to be trying to minimize the use of fertilizers and pesticides. So this is where we're trying to swap some of those out for more natural techniques like crop rotation, like using manure, using biological control methods. With biological control, what we're doing there is we're sort of swapping out the pesticides for a natural predator. So rather than using a chemical, we get the natural predator of our pest species, and we then let them just basically eat the pest instead. Downside is that we are talking about a living thing. They don't always do what we want them to do. They may decide to leave the area. Problem. They also may eat other organisms and just leave the pest quite happily munching on your crops as well. So they're not perfect. Some other things that we can do to try to obviously meet the demands of our population is things like fish farming. So we know that fish are a really valuable source of protein, but overfishing has actually led to these reductions of populations of certain species. So what we've introduced are fishing quotas that limit the number and types of fish that can be caught in any given area, and that gives those populations a chance to recover. We can also use a fish farm where the fish are kept in cages in the rivers or the seas, and that protects them from predators first of all, and when you want the fish, they're right there. It's easy to get. Now, that will again allow those wild populations to recover. A couple of downsides though. First of all, disease is going to spread quickly because they're close together. And secondly, those diseases could also spread to the wild populations. Another technique we can use is something called hydroponics, which is where we're not growing plants in soil. Instead, we're growing them in a solution that contains all the minerals that they need. So because we're not growing them in soil, you can stack them one on top of the other and just have all these layers of plants growing in a smaller ground area. So this basically is all about maximizing space. So we're not growing them in soil, we're growing them in a solution that has all the minerals they need, and it also has the bonus of reducing the chance of diseases spreading, because a lot of diseases in plant come from the soil. Back for everyone, combined and biology higher foundation as we go on to selective breeding now. So selective breeding is quite simply how we're going to breed animals or plants together for a particular characteristic. So we might be selecting for high yield, lots of wool, whatever it is. So the way we do this is we identify the characteristic that we want, we select the parents with high levels of that characteristic, we breed them together, select the best offspring, breed them together, and keep going. And over many, many generations of this selecting for the characteristic and breeding those together, we eventually end up with that characteristic in large numbers. There are some downsides to selective breeding though. It's going to reduce variation within the gene pool first of all, because we're reducing the number of alleles present if we're constantly selecting for certain ones. That means they're going to be more susceptible to disease. And the last one is that it's also going to increase the risk of inheriting a genetic disease. A lot of genetic diseases are recessive, so they need both alleles in order to show. And if we're breeding from a relatively small gene pool, the chance of that is going to increase. So we see more genetic diseases occurring. Now, the next process is one that's a little bit more high-tech, which is genetic engineering. Now, genetic engineering is where we're altering an organism's genome by selecting genes that create a certain characteristic. So we're changing both the genotype and the phenotype here. So good examples of where we carried out genetic engineering are in things like cotton to increase the crop yield, in corn where they then produce the toxin naturally to kill the insects, or in bacteria where we can use them to produce drugs for us like insulin. So the way that we're actually going to do this is we take a foreign gene, so a gene from one organism, and we then insert it into plant or animal cells at an early stage of development of the one we want to show that characteristic. For those of you doing GCSE biology, you need to know a little bit about the risks here. So when we're looking at risks, we've got a bit of an unknown. We don't know the long-term effects. And some of these negative effects might be health problems because we're introducing new allergens to food that weren't naturally there. We could see cross-pollination with wild plants occurring, which could disrupt the balance in our ecosystem. And other people just believe this is unethical because we're interfering with nature. If you're doing the higher tier, combined or GCSE biology, you need to know how we create these genetically engineered organisms. So 3 key steps here. First one is we identify the gene for that characteristic. Second, we remove the gene from the donor organism. And third, we insert it into the host organism. That's the overview. We need to know a little bit more detail. Now, the way we do this is through the use of enzymes. So once we've identified the gene we want, we use an enzyme called a restriction enzyme. And that's going to cut the DNA at a particular base sequence. We use the same restriction enzyme to cut the actual plasmid, if we're talking about bacteria here, to actually cut that plasmid in exactly the same base sequence. So now what we've got on our gene and on our plasmid are these complementary sequences because it's cut it at the same point. So when we then mix our gene with our plasmid and with another enzyme called DNA ligase, then it joins it together. And as a result of that, we've then joined our gene for our particular characteristic with the plasmid. We can reinsert the plasmid into our bacteria, and then it's got the human gene in there. We can allow that to replicate as bacteria do. Every bacteria that comes from it will have that gene, and we can then just siphon off the useful substance, whether it be a hormone or an antibiotic, whatever it was, we can now just collect that. The reason that we use those restriction enzymes, as we said, is they cut the DNA at a specific base sequence, and it leaves that complementary sequence on the gene and the plasmid, and it's called a sticky end. So basically, they kind of stick out and they're going to join together. Now, what we do with the ligase enzyme is that's going to join the donor gene and the plasmid by joining the sticky ends through the formation of a bond. We then have complementary base pairing between the complementary bases by another enzyme whose name you don't need to know. Once we've done this, we're not just going to sit and think, perfect, it's done every time. We need to double check that it's actually worked. So in order to do this, what we use is something called a marker gene. And there are 2 types of marker gene we could use, either antibiotic resistance genes or fluorescence genes. So if we're going to be looking at the antibiotic resistance gene, first of all, you insert your marker gene into the plasmid at the same time as the gene for your desired characteristic. We then transfer the bacteria onto an agar plate that contains that antibiotic. We incubate it, allow it time to grow. And then when you take that plate out, the colonies that are present will all have the marker gene present. And because it was introduced at the same time as our actual desired characteristic gene, they should have that too. So we select those and we can then produce them in large quantities. Very similar process with the fluorescent marker gene. Again, inserted at the same time as our desired characteristic gene, transferred to an agar plate, incubated, allowed to grow, and to check the results, we shine a UV light on it, and if they glow, they've got the genes. We select those ones. Everyone again, so combined, biology, foundation, higher, as we have a look at biotechnology in farming. Now, biotechnology is where we're going to use biological processes or living organisms to manufacture products. And we're looking at genetically modified organisms, or GMOs. 2 GM crops that are very common these days are golden rice, first of all, where we've taken a gene from daffodils and put it into a rice plant so that the rice then produces beta-carotene, which we need in our body to make vitamin A, and that helps stave off blindness in those people who don't actually have access to a wide range of foods in their diet. And we've also got BT corn, which has got the gene from a bacteria, which has been inserted into a maize plant, and that then codes for a protein that's poisonous to insects. So it's like built-in pesticide. For those of you doing the higher tier, you need to know about how we do this. And one of the key words we're going to use here is the word vector, which is basically something that's going to transfer foreign DNA to an organism. And the vector could be bacteria, it could be a virus, it could be a plasmid, but it's going to transfer foreign DNA to another organism here. So what we actually do is we identify the gene that's going to produce this useful characteristic first of all. And once we've got that gene, we're going to use restriction enzymes to cut it. We then use our vector, and using the same restriction enzymes, we cut that DNA. mix the two together, and what we've then got is our desired characteristic gene with our vector DNA. Once we've got it in the vector, we can then go through a process called insertion, whereby it's going to insert that into the plant cell, for example. It then gets incorporated into the host DNA, and then that gene will be produced in all of the organism. So what we've then got is our transgenic organism. So that is all of that bit. And the last bit we now need to come on to is for everyone again, which is all about the health and disease. So when we're thinking about health and disease, what we're looking at are two categories of disease: communicable and non-communicable disease. So communicable diseases spread between organisms, non-communicable not spread between organisms. If we've got a microorganism causing disease, that's what we refer to as a pathogen. Now, there are 4 types of pathogen we need to know about: fungi, bacteria, virus, and protozoa. You need to know examples of animal and plant diseases for each. So for the fungi, first of all, in animals, we've got athlete's foot, and in plants, we've got powdery mildew. For bacteria, animals is tuberculosis and crown gall disease in plants. For the viral disease, in animals is influenza, and plants is tobacco mosaic virus, or TMV. And for the protozoa, we've got malaria in animals and coffee phloem necrosis in our plants. Now, what we actually need to understand there is that those are all caused by something being spread from one to another. So they're all communicable. So they're spread from one organism to another. The non-communicable diseases are the ones that don't spread between organisms. So these are going to be caused by things like having a poor diet. It could be caused by obesity, it could be caused by a genetic disorder, or it could be caused by something in your body not working right. So they're not going to spread from person to person, but they're still a disease all the same. What we do find, though, is that certain communicable and non-communicable diseases have separate causes, but the conditions are linked. So, for example, HPV, which is human papillomavirus, will cause most cases of cervical cancer. We've got HIV, which causes AIDS, and that's going to weaken your immune system, and then it leads to easier infection from things like TB, for example. In terms of how communicable diseases are spread then, we've got a few key ways. First ways that we can have are cuts in the skin. So if you've actually got some kind of a cut on your physical barrier of your skin, infection can occur. Through your digestive system, respiratory system, or reproductive system are other key ways. One of the ways it's going to get into your respiratory system is through something called droplet infection. So at the moment, people may well have coughs and colds around you, and if they're gross enough to not cover their mouth and nose when they actually sneeze and cough, then every time they're doing that, then they're projecting these droplets from their mouth and nose. And in each of those droplets, there will be these pathogens. So what's going to happen is, as it's being projected at high speed from that person, it's then going to splatter on all of you sitting around them, the desk, books, whatever else is around, and potentially infect you. Hence why cover up. If we're looking at the pathogens that are able to spread between plants, then they're doing so through the soil and the water. Now, in addition to this, we can also see things being spread by vectors like insects. So just bear in mind, it's not just soil and water for plants. Insects can also play a role here. When we're looking at a communicable disease, it's not a case of as soon as someone coughs on you, then you are instantly ill. Because what we have is something called the incubation period, which is the time between you becoming infected and actually becoming ill and showing those symptoms. So during that incubation period is when the pathogens are reproducing. And as they're going through that process of reproduction, they cause cell damage or they release toxins. Now, hopefully you remember that bacteria are going to replicate through a process called binary fission. So we start with one bacteria, they replicate their DNA, split into 2, and so on. So they're doubling in number in each stage there. So they demonstrate something called exponential growth. So we start with quite a small number, and very quickly we get a very high number. If we think about a virus, then they can only reproduce by taking over the host cells and using them to generate new viruses. So what we find is the virus infects a cell, it inserts its DNA into the host cell itself. That will then take over the host cell machinery, use it to make more virus, and then finally it bursts out through the cell, releasing all those new virus particles. When we consider how we can prevent this delightful spread of all these diseases, then some of it's just basic hygiene and manners. So covering your mouth and nose when you're sneezing and coughing. Obviously, not sharing needles. So if someone's used a needle, you don't just go, oh yeah, I'll stick that in myself too. Making sure that you're drinking clean water. Don't go and drink from a random puddle, for example. If you're traveling somewhere where they have things like rabies, then don't go up and poke the local wildlife, because if they bite you, you get rabies too. Obviously, making sure you're cooking your food properly, washing your hands, particularly after you've gone to a toilet, using protection in sex, okay? If someone does have an illness, don't go and then stroke them or lick them or anything weird like that. Those are pretty basic things we can do to avoid getting infected with these diseases. If we think about our farmers identifying a disease, then if it's talking about plants, we can obviously take the plants, burn the diseased material to avoid that spread. Saw with the foot and mouth outbreak a few years ago. We do need to know a little bit more about our delightful infections now. So, athlete's foot is our first one, which is caused by that parasitic fungi. The ideal conditions for athlete's foot are those warm and humid areas. So between your toes is a favourite here. And what's going to happen is you get this cracked, flaking, itchy skin as a result. Now, this is going to spread through either direct or indirect contact, so either playing footsie with someone who's got athlete's foot or walking on a surface they've already walked on barefoot. But it's easy to treat. You just get an antifungal cream, apply it, clears it up nice and easily. Next one is food poisoning, which is caused by bacteria that create toxins, and 3 good examples of this: Campylobacter, Salmonella, and E. coli. Now, these are actually capable of surviving both freezing and refrigeration. So if you've got food that's actually infected with them, putting it in a fridge isn't going to kill it. It's just going to slow down its reproduction. The only way you kill it is by cooking your food thoroughly. If you're unlucky enough to get food poisoning, then you can expect stomach pain, diarrhea, vomiting, simultaneously, sometimes explosive, a fever, and if it's incredibly serious, it can lead to death as well. So obviously the spread here is mainly through poor food hygiene, and we treat it with fluids usually. Next type of infection are the STIs, sexually transmitted infections, caused by a range of different bacteria and viruses, a whole host that you can pick from here. And obviously the spread here, as the name suggests, is person to person through unprotected sex or genital contact. So we can spread it through the bodily fluids or skin on skin. The downsides to some of these infections is that they are asymptomatic, so they don't show symptoms when you're infected. So that means you might be infected and just not know it, which means you're more likely to spread it to someone else. In terms of how you can avoid it, don't have sex, most obvious one, or just use a barrier method of protection like condoms. Some key STIs then for you: chlamydia, always a fun one, caused by bacteria there. Pain when urinating and discharge from the penis or vagina. Treat it with antibiotics because it's a bacterial infection. Gonorrhea, another bacterial infection. Burning pain when urinating, vaginal discharge, again, antibiotic treatment. Genital herpes is a viral infection this time, where you get painful blisters or sores on the genitals, as the name suggests, and there is no cure for this one. So if you get genital herpes, you are going to have genital herpes for life. So it's that gift that keeps on giving and just recurs every once in a while just to have fun. HIV is our last one, caused by a virus. Now, the symptoms of this one, you get your weakened immune system, and it often results in AIDS. There's no cure, but we can control the symptoms using antiretroviral drugs, but you have to take them for the rest of your life. HIV itself is human immunodeficiency virus, and this is a particularly tricky virus because it invades your white blood cells, which are the parts of your body that are designed to actually fight infections. So it's a sneaky little virus in the fact that it hides inside the cells designed to actually attack them. And it's reproducing inside those white blood cells. So one thing is going to prevent them from producing the antibodies, so you can't fight that infection. And as it then bursts out of the white blood cells, once it's reproduced in there, what we then find is it's reducing the number of your white blood cells. So it's less effective as an immune system. That means you're more susceptible to common infections, and you're more likely to then die of something that a healthy person would be able to fight off. In terms of our plant diseases, tobacco mosaic virus, first of all, is a viral infection, as the name suggests, and we get these mottled or discoloured leaves as a result of that virus preventing chloroplasts from forming. So that means the plant's not going to grow too well, it's got very stunted growth and lower yields. To prevent the spread, just remove infected plants, wash your hands and equipment between use, and then plant TMV-resistant crops the next year. Crown gall disease is the bacterial infection, and this one's going to enter through a wound on the plant. It integrates into the plasmid, and therefore it creates greater growth chemical production. Now, what we get are these things called galls, as the name suggests, with crown gall disease, which are these growths on the actual plant itself. So it's like a tumour on the plant. Now, they could actually encircle the entire trunk or stem, which would cut off the flow of sap, leading to stunted growth and death again. In order to prevent the spread, we destroy the infected plants and avoid planting any susceptible plants in that area for 2 years. Fungal disease in our plants is powdery mildew, and it produces, as the name suggests, a white powdery spots on the leaves. Now, this is going to reduce the growth of our plant, and potentially leaves drop early, reducing the yield of our plant again. This one's going to be spread where we've got high humidity and moderate temperatures, and it survives on plant residues between the different planting seasons. And the way that it's going to spread is by releasing these spores, as a lot of fungi do, and they get spread through the wind to other plants. So we can treat it by using a fungicide, which kills fungi, as the name suggests. For those of you doing GCSE biology, you need to know about the defences that our plants have. Two types. We've got the physical defences, which are the physical barriers to stop those microorganisms entering, and the chemical defences, which are where we're secreting a substance in order to kill the microorganisms. Physical defences, first of all, most common one is the cuticle, which is that waxy substance that covers the epidermal parts of our plant. It's going to reduce the water loss, and it also prevents the pathogens coming into contact with the epidermal cells, reducing the chance of infection. Now, it's also waxy, and waxy substances don't generally let water sit on them. It makes it run off because they're hydrophobic. What we find is, within the water, we quite often find fungal spores. So if the water's not sitting on the top of the plant because that wax makes it run off, then the fungal spores don't sit on there and they don't germinate on the plant. We've also got the cell wall, which is a major defense against fungal and bacterial pathogens because it's a brilliant structural barrier. Now, all plants have a primary cell wall, which provides that structural support, and it will also, as I say, make this barrier between the actual cell itself and the outside world. It's made of cellulose in our plants, hopefully you remember that, and those cellulose fibres are actually cross-linked with one another and other substances like pectin. So that forms this gel, which helps then cement the neighbouring cells together, so they form this barrier. In terms of the chemical defences, we've got a few different chemicals that plants could produce in order to help protect themselves. We've got citronella and pine resin, which repel insects, and you use those anyway on any of those little insect bands you get on holiday. The pyrethrins, which are insecticides, kill insects. Phenols, which will disrupt the bacterial cell wall, and defensins disrupt the cell membrane, so they're both antibacterial compounds. We've got antifungal compounds in the form of chitinases, which break down the cell wall in fungus, which is chitin. And we've also got caffeine, which is toxic to fungi and insects. And then finally, we've got cyanide, which is just toxic to pretty much any living thing you can imagine. If you're doing GCSE biology higher tier, you need to know how we identify plant diseases. So in the field, we've got two key techniques, observation and microscopy. So observation is looking at the visual symptoms, so discoloured leaves, etc., and microscopy to identify the pathogen, usually through the use of a light microscope. Downside to these field techniques is we're usually waiting for the symptoms before we can identify it. So it's not as quick, and therefore the treatment isn't always as effective. We could also use lab techniques, DNA analysis and antigen identification. Now, if we're talking about DNA analysis, this is where we're going to end up running our little sample through a gel. Now, what we end up with are some lines. So if we've got our sample here and then our known bands of two particular pathogens, what we do, you match the bands. So our sample matches this one. So this is the pathogen that was infecting our plant. That's our DNA analysis. The antigen identification is our second one that we can do here. And this one is looking at the protein spikes, which are the antigens, on the surface of our microorganism. And if we identify the antigen, we've identified our pathogen, because specific antigens are found on specific pathogens. And that's through a chemical analysis. Back to everyone. So combined higher foundation, higher, plus biology foundation and higher, so we can have a look at the blood and body defense mechanisms. Hopefully we know that if you cut yourself, it's not a never-ending wound. You will eventually heal, and the first step of that is to produce a scab. So what actually happens is when you've cut yourself, then platelets, which are one of those main components of your blood, change the chemical fibrinogen into fibrin. And this fibrin then forms this network of fibers like a mesh. The red blood cells then get trapped within there and forms this blood clot. The clot then hardens, and that's what your scab actually is. Obviously, the big advantage of having that scab means that you've then no longer got an open wound, so pathogens are less likely to get into your body. When we're looking at the defense mechanisms, we do have what are called non-specific responses, first of all. So these are the ones where it's just preventing the entry of all microorganisms. It's not specific to any given one, it's just the whole shebang. So things like your skin as a physical barrier, it just makes any of these microorganisms have a challenge to get inside, because you've got all these dry, dead cells on the surface of your body, which make it hard for them to get into the living cells. You've got sweat glands on your skin that produce oils that can kill the microorganisms. We've got things like stomach acid, which kills the pathogens on contaminated food and drink. You've got the cilia and mucus in your airways. The mucus traps the microorganisms, and the cilia are the little hairs that then waft it up to the top of your throat so you can then swallow it down, so the bacteria are then destroyed in the stomach acid. You've got tears, which contain lysozymes, which will destroy bacteria. They're not specific to any type, they just do the same thing with any microorganism, hence non-specific. Then we come on to our other type of defense, which involves our white blood cells. Two types of white blood cells in your body. You've got the phagocytes, which are the ones that will engulf and digest in the process of phagocytosis. And we've got the lymphocytes, which are the ones that produce antibodies or antitoxins. So do remember that when we're talking about the phagocytes, then when we've got a pathogen present, all they actually do is they're going to come in, and if this is our little pathogen, they engulf it, so they surround it, and then they're digesting it with enzymes inside them to break it down into something harmless. The lymphocytes, they're producing an antibody which is specific to the antigen on the surface of our pathogen. Now, an antibody itself is a protein, and those proteins will bind to the antigens on the microorganisms. Once that's occurred, then the pathogen can be ingested by a phagocyte, so we then have phagocytosis. So because we're looking at specific pathogens, then this is a specific form of defense. The good news is that when you've been infected with a pathogen that has actually just one particular antigen on the surface, then what we will do is have antibodies to that particular antigen. Now, some of your white blood cells will remain in your body after this infection has passed as what's called a memory cell. So that if you then get infected by the same pathogen a second time, you've already got white blood cells that know how to make this antibody. So they can respond faster and eliminate the pathogen before you show actually symptoms. And that's what we're referring to with immunity. Those of you doing higher tier biology, you need to know about monoclonal antibodies. Now, a monoclonal antibody is produced in a lab using a type of cell called a hybridoma. A hybridoma, quite simply, is a cell we've made by fusing a myeloma, which is a cancer cell, with a lymphocyte, a white blood cell. Now, they're called monoclonal antibodies because they're produced by a single clone of cells, hence mono, one, clonal, clone of cells. And each monoclonal antibody has been designed to target a specific type of cell. And the way they do that is by binding to antigens of our target cell and killing it or preventing it from operating effectively. So in order to make these, what we do is we start off with our little GM mouse and we inject it with our required antigen. As a result of that antigen being in our mouse, then they produce the antibodies to to that specific antigen. We then collect those lymphocytes that are making the antibody and fuse them with the myeloma cell to make the hybridoma. Now, the reason that we do this is the myeloma cells reproduce indefinitely and the lymphocytes have to be fused to survive outside the body. As those hybridoma cells then reproduce, they form identical clones of one another, and we get a large number of those actual hybridoma cells. Now, each of them is going to produce the required antibody, which we can then just harvest, and that's our monoclonal antibody we can then use. In terms of where we use our monoclonal antibodies, one of the more common ones is in pregnancy testing. So this is where we've got a particular hormone called HCG, human chorionic gonadotrophin, and that's going to be produced from 2 weeks after conception. Now, the little pregnancy test stick itself has got monoclonal antibodies that bind to HCG that are actually on the little stick itself, and they're bound to a dye molecule. So what we then find is that when the pregnant lady wheezes on the stick, then it's actually going to move those dye molecules up to the window where we get that little line, and they then bind to the monoclonal antibodies and stick in this little line that makes your little positive line. If obviously you're not pregnant, doesn't do that, you don't get a line. We can also use monoclonal antibodies to detect disease because they act as markers by binding to specific antigens. So things like prostate cancer we can use here. And we can also use them in treating certain cancers, because using a monoclonal antibody to target that specific cancer cell, then we can attach a certain drug or radioactive substance to it to deliver it right to the tumour itself. Now, obviously, the big advantage of that is it's going to reduce damage to the surrounding tissue and only target the actual tumour. Biology combined, higher foundation, everyone back for the bit on vaccination then. Now, vaccinations, quite simply, are where we've got a small amount of dead or weakened, which is known as attenuated pathogens, which we're then going to somehow get into the body, usually through an injection. Now, once we've inserted that into the body, your lymphocytes will produce antibodies to the pathogen. We get the memory cells, which remain in your body, so that if you then encounter that same pathogen at a later stage, the memory cells replicate quickly, produce the antibodies before the symptoms show. So you've got immunity to it. Another kind of chemical that we can use is an antiseptic. Now, antiseptics are the ones we use on external living surfaces to kill or neutralize pathogens, but not damage the human tissue. So this is things like alcohol and iodine. So when someone goes into surgery, for example, they'll use something like iodine on the skin, first of all, to kill any of the pathogens on the surface before they cut you open. Now, we do have different antiseptics that act on different microorganisms. And if we're talking about a non-living surface, so if you're looking at wanting to wipe down a desk, then we don't use an antiseptic, we use a disinfectant. Does the same thing, kills any of our pathogens on the surface, but it's a non-living surface. So antiseptics, living surfaces, disinfectants, non-living surfaces. Another chemical we need to know about is the antiviral, which is the drugs that destroy viruses. So many of these are actually specific and designed to act on only one type of virus. And so they can do this by actually blocking the virus entering the host cell. They can prevent the virus releasing genetic material or prevent it inserting its genetic material into the host DNA. So antivirals work on a virus. Antibiotics are the ones that kill bacteria. So don't say that antibiotics are used to treat a viral infection. They're not. Now, we've got several different types of antibiotic which kill a range of different bacteria. And if we ever need to identify the bacteria making someone ill, then we'll take a sample of blood or stool, for example, culture it in the lab on an agar plate, and use antibiotic discs then to identify which antibiotic is best to use. Now, obviously, if you go to the doctor, they're not going to do this for every infection because we've got general antibiotics that are the go-to for certain infections. It's for those cases where we've tried a range of different antibiotics, it's still not working, and we need to find a solution that we would do that. One of the other assessed practicals that you would have done is the zone of inhibition. So this is looking to see the effectiveness of different substances at killing bacteria. So what you have is your Petri dish, which has got a lawn of bacteria spread across it. And then what you're going to do is you're going to place little bits of paper at fixed points, and these bits of paper have been soaked in different chemicals, whether it's antibiotics, whether it might be disinfectants, antiseptics, whatever it is that you want to see how well it kills bacteria on that plate. And then what you do once you've incubated it for a while is you take it out, and we're looking for these clear zones, these zones of inhibition. So we need to work out the area, and because it's a circle, then that's just pi r squared. And that means that we can then work out which is the most effective. The larger the zone of inhibition, the greater the effect at killing the bacteria. For those of you doing GCSE biology, you need to know about aseptic technique. So aseptic technique is just preventing foreign microorganisms from being introduced to our test sample. So we're just ensuring that the apparatus in the environment remains sterile. So it's doing things like washing your working areas with alcohol before and after so that we're not contaminating it. You're wearing gloves if there are any pathogens that could infect you. You're going to autoclave glassware and apparatus to sterilize it. You're going to work close to a Bunsen flame so that nothing falls in from the air. In order to actually carry out aseptic technique to use a Petri dish and transfer samples, we use a little wire loop. Now, what you're going to do to ensure that's sterile is you hold it into a Bunsen flame until it glows red, and that's going to kill anything present on the loop itself. Once it's cooled, you then take your sample, and then you can create things like your streak plate. When we've actually grown bacteria on a plate, then we can identify them. One of the key ways we do this is just by looking at the colony itself. So the shape of it, the colour, what's its edge like? Is it smooth? Is it jaggedy? Does it have a raised surface? Is it smooth surface? Is it a bumpy surface? All of those sort of things we can look at to then allow us to identify the bacteria from the colony. If we can't identify it purely by that, we can also use chemical tests to then give us that last bit of identification. Now, once we've actually identified our bacteria, we'd obviously know how to deal with it and treat it. Back for everyone again, so combined and biology, foundation and higher, we need to know about medications and new drugs. What we generally find is a lot of drugs come from plants, but we've actually created synthetic versions since we extracted them from plants in the first instance. Now, in order to actually develop these, we start off with computer models in order to develop possible drugs that may work. And then once we've got our potential list of chemicals, what we're going to do is then go through a range of different trials to see if it works and if it's going to work safely. So what we've actually got, first of all, are preclinical tests. Now, the preclinical tests will consist of live cells, first of all. Then we could also go on to the animal tests. Now, what we're doing in terms of the live cells or the tissue is we're looking to see when we add this chemical to the cells in a Petri dish, does it kill them all? If the answer to that is yes, then that chemical is not going to work as a drug. Test over. If it doesn't kill them, if it doesn't make them replicate in crazy ways or do odd things that we weren't expecting, we can move on. Assuming it passes those live cell trials, we go on to the animal testing phase. Now, animal testing, the whole idea here is to see what happens when we put it in something that's actually living. Does it make their behavior go insane? Does it speed their heart rate up? Does it make their brain explode? Anything like that that we wouldn't be able to pick out just from some cells in a dish, but we're not going to give it to a human just yet in case it does any of those things. Now, the animal testing has got to pass with 2 species before we can move on from the animal testing phase. Obviously, animal testing is not without its problems because some people may not agree, animals can't give their consent, etc. But we do use the 3 R's principle to try to make it a little bit better here. So reduction, using the smallest number of animals possible. Refinement, improving the experiments to avoid any unnecessary suffering and improve the animal care. And replacement, replacing animals with other techniques where we can. If we've passed the animal trials, then we go on to our clinical trials. So this is where we're on our humans finally. 3 key stages here. First one, healthy volunteers, where we're looking for side effects. So we're not going to give it to people with a disease who are already sick. We're going to give it to healthy people and see what happens there. Second one is a small sample of volunteers with the condition in order to actually see how effective this is. And then our third and final stage is our large group of volunteers with the condition in order to actually test the effectiveness and check the safety. Obviously, at each stage, if we suddenly get some very disturbing side effects, the trial will come to an end. We don't just keep going with it and think, that'll be all right in the long run. It's always about checks and measures, and all of those tests take a long time and cost a lot of money. So we're talking about potentially up to a decade and millions of pounds to develop one single drug. When we're going through the human trials, we also need to consider the placebo effect, which is where people start to feel better because they expect to feel better. Now, what we do is we're going to use something called a double-blind trial to overcome this. So in the double-blind trial, some of the patients in our group are going to get the actual drug we're testing, while others are going to get an exact replica of that drug, but it's got no active ingredients at all. That's our placebo. So it looks like the drug, but it isn't the drug. Now, the key thing about the double-blind trial is neither the patient nor the doctor knows who's getting the placebo and who's getting the real drug. And that just means we've reduced the chance of anyone sitting there and putting any false results in or saying, oh, this person did really feel better because they got the drug that that doctor's trying to develop and therefore sell. So double-blind trials, no one knows. Last bit we need to go on to then are the non-communicable diseases. We are almost there. So first thing we need to consider is smoking. We've got 4 key chemicals here. First one, tar. It's a carcinogen. It's going to cause cancer as it collects in the lungs. Second is your nicotine, which is the addictive drug that's going to affect the nervous system, increasing the heart rate, narrowing blood vessels. Carbon monoxide, which attaches to the hemoglobin irreversibly so you can transport less oxygen. And particulates, which are little tiny pieces of solid that then get engulfed by white blood cells and can weaken your alveoli walls, leading to emphysema. Next problem we've got is alcohol. Now, alcohol contains ethanol, which again affects your nervous system. It's a depressant, so that's going to slow down the reactions within the body. Short-term effects, these are things like blurred vision, loss of balance, slower reaction times, that change in behavior, so that your normal happy-go-lucky friend that just bursts into tears when they've had too much to drink or becomes that aggressive idiot in the corner. Your long-term effects, this is where we've got the more serious ones, where you've had long-term use of alcohol to quite a high level. Cirrhosis of your liver, where basically your liver is degrading to all intents and purposes, stomach ulcers, heart disease, brain damage. And what we're then going to find is that as a result of those, it can lead to death. So not great to consume alcohol in large quantities on a regular basis. One of the biggest problems we've got in terms of a non-communicable disease is cardiovascular disease, which is a disease of the heart or your blood vessels. One of the most common ones we've got is atherosclerosis, which is where your arteries have become hardened and narrowed. So what we end up with normally is your arteries are lovely and clear, so blood can flow through without any issues. With the atherosclerosis, what we end up with are these deposits of fat building up in the walls, which is going to mean there's less space for the blood to flow through, and it also makes it harder for them to expand as they normally do with blood flow. Now, in addition to obviously the buildup of fat in your arteries, you can also have what's called a thrombosis or a blood clot. Now, depending on where that blood clot occurs is going to determine what's going to happen to you. So if you get a blood clot in one of your coronary arteries, which are the ones that supply your heart with the oxygen, etc., you get a heart attack. If it occurs in one of your arteries which supplies your brain, that leads to a stroke. So different regions cause different effects. When we're considering the potential risk factors for CVD, high salt diet is one of the key problems here. If you've got too much salt in your diet, then more water is absorbed back into the blood after being filtered in the kidney, leading to increased blood pressure. Another common one is obviously a high fat diet, particularly those saturated fats, because that causes cholesterol to be deposited in your artery walls, narrows the blood vessels, restricts the blood flow, increases your blood pressure. And we're going to find a lot of that in things like red meat and butter, so very common in our Western diets. In terms of what can we do about this, it's not just a case of, well, you know, get it, what matters, what can we do about it? You can exercise. Obviously, throughout school, we forced you to do PE. When you leave school, no one forces you to do PE, but you need regular exercise to stay healthy. Obviously, taking regular exercise means that you're going to have a lower body mass, and that means that less of the food you eat is stored as fat. That's going to reduce your risk of CVD and also diabetes. If you've got lower body mass, then you're going to have healthier joints and therefore lower your risk of arthritis. You're also going to have a much stronger heart and lower cholesterol levels overall, which is always good. We can also look at some other lifestyle changes to reduce your risk of CVD. Eat less processed food, cook fresh, always the best way. Obviously, reduce your consumption of alcohol where present, and you can stop smoking if you do smoke, because all of those are going to help to reduce your chance of getting cardiovascular disease of any type. If you do get cardiovascular disease, then we can treat it in a few different ways. First one is a medication approach. So we've got 4 different types of medications you need to know about here. Statins, first of all, which reduce the blood cholesterol by preventing its formation in the first place, but these can lead to an upset stomach. We've got antiplatelets, which reduce the stickiness of the platelets, which reduces the chance of blood clots. But if they're reducing the chance of blood clotting, you can develop internal bleeding. Not good. Beta blockers are our third one, and these are going to block the effects of adrenaline, so slows your heart rate and improves blood flow. Can also lead to dizziness and tiredness on the downside. Fourth and final one, the nitrates. These widen your blood vessels by relaxing the blood vessel walls, allowing more blood to flow through at a lower pressure. But again, dizziness and headaches can occur in that one. Next option, if the medications aren't enough, we can go to a surgical approach. So we could replace a faulty valve. So if in your heart one of your valves isn't working properly, we can actually replace that. We could carry out a process called an angioplasty, which is where we insert a stent, which is basically like a little mesh tube that then opens up your arteries and widens them again. We could carry out coronary bypass. We're getting more extreme at this point, as you can tell, where we take a blood vessel from somewhere else in your body, arms, legs, etc., and we're then going to use that to bypass your blocked coronary arteries and reconnect your heart to a blood supply. Or in the most extreme cases, a full-on heart transplant. And obviously not easy to do because you've got to have a tissue match, and hearts are something we don't have a spare of in our bodies. The downsides to obviously having a transplant is if you don't match it, your body will reject it, because what we must have is the tissue type matched to the actual recipient there. Otherwise, your immune system just identifies that's not yours, attacks it, and destroys it. In order to reduce that risk of rejection, then, as we said, we carry out tissue matching first of all, and then you're also going to be on immunosuppressant drugs. So it just reduces the effect of the immune system and makes it less likely you will reject. We've also now got these advances in stem cell treatments. Now, a stem cell is an unspecialized cell, which we usually take from embryos. So we take embryonic stem cells because they can differentiate into any cell type. And one of the most common sources of these are surplus embryos from fertility treatments. Now, in terms of what we use these stem cells for, we can test drugs. We can also potentially reverse damage caused by diseases, things like Parkinson's disease, where we can actually create new brain cells. We could rebuild bone and cartilage in arthritis. We can replace heart valves using them. In terms of the concerns we have, we don't know the long-term effects because we haven't had this technology for that long yet. But there is potentially an increased cancer risk, and we've got the potential for possible rejection of that foreign material in the body again. If we talk about gene therapy, which is again one of these kind of more modern treatments, then this is where we're actually replacing a fully functioning allele for a faulty one. So the faulty one we're getting rid of, and we're putting a fully functioning allele into its place. So this is where we can have a look at cystic fibrosis, which is caused by one faulty CTFR gene. If we replace it with a normal CFTR gene, then we've actually got our functioning protein channel, and we've got rid of that problem. So what we're actually doing here is using restriction enzymes again to cut the normal version of a gene from the DNA of a healthy person. We produce many copies of the normal allele, and we insert it into the allele of the person who has the genetic disorder. Problems with this, though, is the healthy alleles may not go into our target cells, or at least not all of our target cells. The healthy alleles may join chromosomes in a random place and therefore not work as we intended at all. And it's a short-lived treatment because your body replaces those cells, and therefore we lose the healthy allele because it will replace it with your own DNA. We had that human genome project we mentioned back when we did B5, which was all about sequencing the human genome. And we've used that in gene therapy because we know where these genes are that are linked to certain inherited diseases. And we can then hopefully go on to develop drugs that will directly target the disease which is caused by that particular gene or that particular protein. So we can design drugs to target that one gene, that one protein. We could also potentially develop new gene therapy treatments through the use of our Human Genome Project knowledge, and maybe even develop personalized medicines. So they're targeted medications which will have greater success rates and fewer side effects because they're specific to your genome. Obviously, not all of these things are here yet, but we are working on them in the future. And that's it. That is all of B4, 5, and 6, you'll be pleased to know. A little bit longer than I hoped today, so 10 minutes over. Now, our next one is going to be our chemistry exam next week. So I'm hoping to be here for 6 o'clock, but I do need to jump out of a meeting a little bit early. So hopefully my boss will let me. But assuming I'm allowed out of my meeting a little bit early, then I'll be here at 6 to go through the chemistry. And then obviously we'll do the physics as well before your physics exam. So hopefully you found it useful. Do remember tomorrow to read the questions carefully. Remember to give every question an attempt, because an attempt is better than a blank space. And good luck. Hope you all have a